From Microbe TV, this is Office Hours for Wednesday, August 13th, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet where you'll hear science. I'm a virologist for over 40 years. I know a little bit about viruses, but also other things, depending on our expertise. We have a collective expertise here in the group. Welcome, everybody. Welcome, moderators. We have Barb Mack tonight. We have Vanity Nutrition. We have... who else? Les is here. So thank you for being here and moderating, keeping things civil. Because, you know, sometimes Trump likes to come here and make nasty comments. We have to kick him out. Also, thank you all for being here as well. It's always fun to have all of you here to participate and ask your questions. What's happening tonight? So it's just me, no guests, because guests are maybe once a month. I have to think about who the next one would be. And we're going to have some news, a couple of news items here. We are going to have a mini lecture on DNA sequencing. It was requested 2 weeks ago. This is something that I did in my career, so I enjoyed DNA sequencing. So you're going to hear about the beginning to the present of DNA sequencing. Should be fun for those of you who like techniques. And of course, tell us where you are from and we can shout you out. We'll do that for a little bit, and then we'll go into our news. Ryman wants to know about open resources for self-study. I haven't used OpenStax. Local library doesn't have a good biochemistry book. But you could buy a used one. They're very cheap used, right? Because they're all beat up, but they still have the information in it. So what's in this OpenStax? Let's take a look. STAX. Free textbooks online with no catch. I see. You know, it really depends on the textbook, right? So not all textbooks are created equal. My virology textbook isn't there. So you just have to be careful that they're good, because it's not easy to teach right through the mechanism of a textbook, and so you have to be careful. I would buy a reputable biochemistry textbook at low price, right? Well, Linda wants to know what the moderators do. Actually, the moderators already answered. Well, they chat and ask questions like everyone else, but they also see if someone's making nasty comments. Some people do occasionally, and then they just bump them out of here. That's it. Like if people say silly things or nasty things. Yeah, nasty is the way. Melinda's in North Carolina, 82F. So here in New Jersey, it's 25C. We just had some thunderstorms, screwed up all the trains. I didn't know if I was going to make it on time as usual. But here I am. Dax wants to know the status of the 25-26 vaccines. Well, you're going to have vaccines. You're going to have flu, and you're going to have RSV, and you're going to have COVID vaccines. What the CDC advisory committee did was to ban all thimerosal-containing vaccines, which is just a flu vaccine multi-dose vial. And they're going to look at the hepatitis B vaccine, because RFK Jr. doesn't think babies should get vaccinated against HPV because he's a moron. He doesn't know anything. Babies are at risk for hepatitis B virus infection from their mothers and other people, but particularly from their mothers at birth. And so you vaccinate them right away. As soon as they pop out, they're vaccinated. And if you don't, they get Hep B, and it's chronic for their lifetime. because they can't clear it, and that could lead to liver cancer. So the vaccine prevents that, and it's a substantial number of potential infections every year. So we need that. But that's not what you're asking about, I know. You're asking about the respiratory diseases, which I think will remain the same. So here's Vanity. I don't know if you can see her picture, but she's got a new icon there, looking very professional as a veterinarian. Who else do we have here? Is Tom here? They must have missed Tom. Tom is here because Vanity said hello to him. Welcome, Tom. Where is she? Where is he? See, Vanity goes, hi, Tom. But I don't see him. But hello, welcome. Thank you for moderating. 77F on the south shore of Long Island. Rita is in Idaho. 101F. Wow. I saw a very nice graphic on Instagram today. All the U.S. states that have exceeded 100 degrees so far this year, most of them had a very hot summer. Even our lowly New Jersey here was 103. Yeah, it was stormy when I left. It was just raining. And then on the train, it was pouring. But then when I got to my stop, it had really subsided. Yeah. I mean, I have a picture. Actually, I could show you, which is actually a nice picture I took. Let me screen share, drop it. And you can see what the rainy streets of New York look like. This is when I just airdrop. No, I don't want it to there. I want it to the other. Oh, that's because I don't have Wi-Fi on. Let's turn on the Wi-Fi. Here we go, Mac Studio. And show in Finder. And we will open it up. I don't need to open it, actually. I can just pop it in here, and it will obscure everything. So I just stepped out of the incubator tonight. Here you go. Can I get it smaller? Apparently not. And that's what it looked like. Yeah, everybody's got umbrellas. You can tell what's going on there. Raining, yes. Yeah, there's Tom. He was way at the beginning. I missed him. Oregon Coast Range, 26C, severe drought. Welcome. Thank you for moderating. Viruses. Luna is in the land of opportunities, Iowa. March 716 is in Amelia County, Virginia. Distant thunder, barely audible. Well, that's the beginning of a poem. Distant thunder, barely audible. It's not quite a haiku, but we could work on it. Right? Distant thunder, barely audible. Those are 2 lines. Wait, a haiku is 575, right? Distant thunder, that's only 4. Barely audible. Yeah, we got to add more syllables. But I like distant thunder. I could just make a friggin' poem. The hell with haiku. Distant thunder, barely audible, loud chirping and chittering. Whole bunch of bugs. Something like that. You wrote a poem, Marge. It's good. I like it. Amelia County, Virginia. Rima is here tonight from Iowa. That's Viruses Luna. I felt bad that I forgot her name a few weeks ago, so I've been practicing. Yeah, I'm very sorry about that. We don't like to diss people by forgetting their name. I think that's bad. Yeah, people forget my name a lot, too. It's okay. Kang, hello from Chicago, 25C with clear skies. Visto is in Sydney. 14C and a bit cloudy. Bird Carver is in Grand Manan Island. We're at 63F. Leo is on the Mendocino coast. 13C. Welcome. Privyet. Veronika from Moscow. Already getting autumn vibes. 13C. Yeah, that's an autumn vibe. Reminder for everyone who loves stars, it's that time of the year with the most falling stars. Laura is in Washington, PA, 26C. Peter is in Boulder, Colorado. Coralie is in New South Wales, near Canberra. Peter is in Boulder, 33C. Jan is in Tucson, 38C. That's very warm, 101F. Wow. Very warm. Veronika. Finally started listening to If Books Could Kill. Their episodes on COVID are great indeed, and they mentioned TWIV. Mike and Lori are in Burien, Washington, 22C, and partly cloudy. Elizabeth is in West Virginia. Bring on the facts. We deal only in facts here. No misinformation. I may make mistakes sometime. That would be misinformation, but try not to do it so much. No disinformation, which is intentionally telling you wrong stuff, like what RFK Jr. does all the time, tells you wrong stuff. Right here, and, here's our Wednesday night opportunity to ask me questions, but we have other podcasts too at Microbe TV, where you can learn science from all different experts. Patricia is in D.C., steamy and humid, with a bit of rain. Les wants to know if we could get Dr. Wilson from Debunk on Livestream. Well, I could ask him. He's been on TWIV twice, I believe. The live stream is 8 to 10. He's got a kid he's got to care for at night, so I don't know if he can do that, but I will ask him. I'm happy to ask him. Then you could ask him to debunk people. Logotic is in Chicago, 25C. Angry Penguin, north of Toronto, 23C, and finally had I don't know. I'll be in Toronto in November for ASTMH, American Society for Tropical Medicine and Hygiene. Rita is in Idaho, 38C, but it's a dry heat. Patricia is from Massachusetts. This is the Nerd House. Yep, I think it is. EHS Talent says, I really think Paul Offit has the right idea. We need to create emotional content showing the pain people feel when a child dies from a vaccine-preventable disease. Yeah, he mentioned that on a recent Beyond the Noise. You can't just give numbers. People don't care about numbers. But if you say, my daughter died of measles, and that's it, then you will appeal to people. And EHS thinks it has to be one of us. Is there anyone here who has documentary making skills? There you go. There you go. Oh, here's the completion of Angry Penguin. It's 23C, and we finally had some rain last night. So some rain last night. There you go. Thank you. Cynthia's in Ventura County, California. 7 degrees Fahrenheit. So something's missing there, too. Camarillo, California. Steel Rabbit is in Vancouver, where it's 78F. Logotic said, Paul Offit just appeared on the Bulwark. I don't know how to pronounce it, because that's not a word I use very often. Bulwark. I guess Bulwark. Reacting to RFK Jr.'s BS, it was glorious. Truth or Dare is in La Jolla, California. Abdul Rahman is in Brooklyn. The lightning is a good background effect. Rob, thank you for your support of science communication. By the way, folks, hit the like button. There are 100 of you in this room and only 40 likes. Are you sitting on your hands? Please hit the like button so maybe we can attract people in the next 2 hours or so, which will zip by in no time. So don't feel bad. Just pay attention, chat. You'll be fine. Long live the incubator. Yes, long live it. I would like it to be long-lived. Micro TV with respect to the status of vaccines, I think it remains to be seen if those under 65 can get COVID vaccines. Yes, and pregnant women as well, right? The, Prasad Vinay Prasad has said no COVID vaccines for under 65 and pregnant women. So, I mean, the vaccines are still available. The question is whether you would, if you wanted to get one and you're in those two groups, if you had to pay for it yourself. It's not clear. But there will be vaccines of those sorts, yeah. Melinda wants to know how bad the flu season is this winter for the Southern Hemisphere. Let's look it up. Flu season, Southern Hemisphere. So what is it? Right now it's winter down there, right? The CDC has an article, but it's from a year ago. 2024 flu season was similar to previous flu seasons. Yeah, that's all from last year. So I don't find anything that's for this season. So maybe the data aren't in yet. Sometimes it takes a while for that to come in. Although they're like our guest here. What's his name? It's the Italian name in Australia. Anyway, he would visto tutti, saw everything. He may know anecdotally how the flu season is, right? Lise is here from Columbus. Great weather this with afternoon rain for my garden. $4:58. I think maybe that's when Tom arrived. I don't know. 4 minutes and 58 seconds in. I don't know. Angry Penguin. Listen to the TWIV last night where you discussed respiratory infections reactivating dormant cancer cells. Very interesting. I learned so much in that episode that I didn't know. I didn't know there were dormant cancer cells, right? This is for breast cancer. So what happens is you're in remission, but there are already metastases to the lungs and bone marrow and one other place, and they go dormant. They don't divide. And they can stay there for many years, and suddenly they start dividing, and then you have a relapse, right? As bad because you have a lot of Mets, you're kind of in trouble. But they found that respiratory virus infections, particularly influenza and SARS-CoV-2 in a mouse model, would reactivate these lung metastases. And then they looked at human data during the COVID pandemic, and they find there was an increase in overall cancers during COVID and people who got infected, and also breast cancers. So it's a real problem. Yeah. I reached my stand goal, Vincent. Nicely done. It's so nice to be complimented by my watch. Julie S. is in central Pennsylvania where it's hot and dry. All the rain missed my area. Here's a good one. Today's the 112th birthday of stainless steel. So I learned today that it's a good factoid. Now I got to remember the numbers. Who did I send it to? Did I send it to anyone? You have 4 trillion cells in your body, and each one of them is doing 2 to 3 billion chemical reactions per second. Can you imagine all that activity? And it's coordinated. No wonder things screw up, right? No wonder we get sick because it's so complicated. I'm reading a really good article in the New Yorker. It's the one with the Statue of Liberty on the cover that was removed from the Smithsonian, right? Ridiculous. And it's all about longevity research, and that's where I got that from. A lot of other great tidbits in that article as well. I had meant to bring it and read some of them out, so I'll do it next week because it's really good. You know, the people who are doing the longevity thing, they're all rich. It's ridiculous. You can't afford it because you got to do a lot of exercises and diagnostics, and you got to take supplements. But as far as I can tell, if you just eat properly, get good sleep, and exercise, you can extend your lifespan. That's what it's about. It's extending your lifespan. But one guy said, you know, just stay healthy, and that's the best. Well, someone else said you have to love too. That's part of it as well. So here's Vanity. Well, you're not going to see probably. That's her dog dressed in scrubs. GSD Bio is in Santa Barbara where it's 20F. Cynthia is in Camarillo, California, halfway between LA and Santa Barbara where it's 73. And Cynthia is a retired special education science teacher. Welcome, Cynthia. Carol, our favorite nephrologist from Bakersfield, where it's 102F39C. Richard is in NorCal, 95 and sunny. Steve is in Columbus, Ohio. And Les has put a link to Paul Offit on the Bulwark. There you go. That Barb liked the street view. Yeah. Isn't that cool? I walked right out. So that's where the incubator is. 7th Ave between 30th and 29th. And I just came out the door, turned left, snap. I just wanted to get and it's got a nice atmospheric feel to it because it just was raining, right? Yeah, Veronica likes the episode and she's commented enough to get a crown. You're the number 1 commenter so far. Well, that's probably a while ago. Let's see now. Because I have 2 sources of, Veronica's number 3 now. I have 2 sources of comments, a little laptop here, and then the main source that I click on. But I'm always behind, right? Jan Lasko is in Tampere, Finland. 9.5C balmy. Oh, yeah. Well, the Finns love to freeze, right? That's why they have saunas. MP is in Ann Arbor, Michigan. Anyone else from Ann Arbor? Douglas, Redondo Beach, 22C. Surfs Up. Surfs Up. Great song by the Beach Boys. No, it wasn't Surfs Up, was it? Maybe it was. Surfs Up. Let's see. This is an album. And it must have been an eponymous track. No, there wasn't. Just an album name. Did you like eponymous? Cool. Truth or Dare wants a comment on, or do a future mini lecture about CCR5 Delta 32. So here's the thing about this. About 14% of the human population, mostly in the Mediterranean area, have, of course, people have since moved, but that's where it seemed to have originated. Some people lack a piece of the gene encoding CCR5, so it's called Delta 32 is the mutation. And so these people don't make CCR5, which is a co-receptor for HIV. So these people are resistant to HIV infection. So the question is, why is it in people? It's been in people a long time, and there was an idea a while ago that it may have been selected by plague. Maybe it protects people from plague. But as far as I know, that didn't pan out, although I can look into it. So we don't know what's selected for Delta 32, right? Something in 14% of humanity is a lot of people. And why is it selected for? We don't know. Well, so some pathogen somewhere must have selected for it, or it conferred viability or greater fitness in some disease condition, maybe. We don't know. We don't know. Linda writes, after they discovered ulcers are caused by H. pylori, it seems more and more diseases have a microbial cause or contribution. Many diseases have a microbial cause. H. pylori was actually down the line when many other bacterial diseases had already been sorted out. But you know how that was figured out, right? The fellow in Australia who was onto this, he actually drank some H. pylori and got an ulcer. He got a Nobel Prize because he was the one who proved that H. pylori causes ulcers. And you can take care of them with antibiotics, right? But they also cause stomach cancer if they're allowed to fester for many years. So you got to be careful there. You got to be careful. Vanity, speaking of haikus, when that phrase came out to uncover bots on Twitter, I used to tell them to write haikus about various things, slugs, toenails, kale, etc. Well, that one I just talked about, it's got the makings of a haiku. It's really good. David is in Dayton, Ohio. Matt is in Phoenix, 41C. Tony is in Long Island. Appreciate your breaking down the paper on the inflammatory response and awakening of cancer. I mean, that's the kind of paper I think listeners love, right? It's mechanistic and it's relevant, right? So I don't know if it applies to other cancers, but if you've had breast cancer and you're in remission, you better watch out for a respiratory infection. You better mask up in the winter among a lot of people, okay? Now, the numbers are not huge in people, but it's an increase, and you don't want to have an infection and then a year later have metastases, right? No. Of course, probably if you get vaccinated, it will reduce the severity of disease, and that may reduce the inflammation and therefore reduce the awakening of these cancer cells. But they didn't look at that in the paper. It's a good question. Tim writes, they've done great work on vaccines for things like HPV and RSV. Is anyone working on anything for RFK? I think at NIH, some people who haven't been fired are still working on what they were working on before. But as far as I can tell, he's not working on it. He doesn't have people working on anything. He's just lying when he says that. He said, he said recently, what was it? What was it that he said? He said he's working on a universal vaccine. And listen to this, what he said. It's going to take care of all viruses in the phylum. The phylum? Really? Do you know what a phylum is? Kingdom phylum. We don't use phyla for viruses. We have other words. Anyway, the technology that he's proposing is ancient. It's not going to do anything. He says we're going to make vaccines better than the ones we have now because, and he defunded the BARDA for mRNA vaccines because he said they don't really protect. And it's just a lie. It's just a lie. So he says we're working on this. I don't believe it. If they're working on it, it's a waste of your taxpayers' money. Nerd House Forever. Hello, Questella. You've been reading the textbooks? Shane Crotty interview with Cindy was great. I'm glad you liked it. Cindy wants to do interviews, which is great. And she'll do it at her place at Cornell and when she travels to meetings as well. She wants to do more of that. Yep. Erie Ozone in Asheville, North Carolina, 75S. Are you going? Jay Shuri writes, are you going to take another COVID booster this year? No, I'm not going to take another COVID booster. I took one when, I don't know when it's the last one I had. I think I had 3 or 4, but I haven't had one in a while. My philosophy is I'm going to try to avoid getting infected by wearing a mask a lot among people. Excuse me. If I get infected, I'll take Paxlovid. Rita says Idaho has its first measles case in an unvaccinated child. Now, Rita, you had said last week that it was found in wastewater, right? So I guess it was just meant somebody was infected, right? Michael, did you ever formally study broadcasting or did just work out your own radio-like style as you podcasted more and more? No, I never studied broadcasting, but thank you for saying I have a radio-like style. Yeah, basically I started podcasting in 2008, but I'd been a lecturer for many years before that, and I knew I had learned how to pace my talk, like just, pause between certain words and don't talk too fast and all of that stuff. So I think that naturally lent me to do podcasting. But certainly over the years, I've slowed down even more. I really believe in the pause. I also think you should tell stories, which is a good part of radio, right? But thank you very much. I'm glad you like it. Maybe it says, Idaho vaccination rates are the lowest of the 50 states in time for back to school. Yeah, that's going to be a problem, right? Going to have big outbreaks. We do have a very active chat in this live stream. We have 140 people and lots of comments. I'm very proud of all of you for coming back and participating. A lot of people go to a chat and they're silent. But here you don't have to worry. If you ask any question, I will attempt to answer it, and I will never say it's a stupid question because that's not how you teach people, right? Eliza, just getting home from my garden with loads of veggies. Southern Ontario. All right. Visto says, anecdote. In Australia, I see lots of people I know getting flu. So that's good to know, but is it more severe or less severe as last season? But you wouldn't have any way of knowing because, you probably know 20, 30 people, right? It's not enough. I've not fallen to the flu yet. Silly me missed my chance for free flu shot 2 months ago. Rita, disseminated dormant cancer cells. This scares me. Yeah, I think it's pretty scary, especially they can be reactivated by respiratory infections, which are really common. It's scary, yeah. Luther saw the movie Mads. It is a rabies-like virus transmission between drug addicts. Interesting. It doesn't happen in real life, of course. Patrick, 2025 flu season update. In the southern hemisphere, flu activity decreased overall, although with different trends. Okay. Yeah, the winter is wrapping up, just like our summer is wrapping up. But it doesn't tell us anything about the severity. Or maybe here. Influenza activity remained elevated in Oceania and some countries in Southeast Asia, tropical and temperate South America. Compared with the previous week, most countries reported stable activity. Slight increase in one country in Eastern Africa. Thank you, Patrick, for looking that up for us. Really appreciate it. MN loves the Frank Zappa quotes. Yeah, the one about the universe being made up of so many neutrons and protons and 85% morons or 95% morons. It's great. He was a smart guy and a good musician, too. Molly is in Hilton Head, 90% humidity. It was 90 degrees. Philip is in Wales. Good to see you, Philip. It's late for you. Appreciate it. Extending health span. Yes, that's exactly what one of the scientists that they interviewed said. Improving your health is great. Improving your lifespan, he said, is effing BS. We don't know how to do that, but we know how to make you healthier, right? Well, we're all going to go for a walk along the Gowanus too this fall. Don't forget. But you guys could walk this summer when it's nice. Yes. Yes. Carol says, eat food, not too much, mostly plants. Yeah. By Michael Pollan, right. That pic was in front of Mustang Sally's. Exactly. Yes. Actually, I just passed it because I don't think we can see it. Let's look. Where's the picture? Yeah, I just passed it because Mustang Sally's is right behind me. It has chairs on the sidewalk. Yep. Yep, it was right past, just past it. How is your dog? She has something like G. Well, she got many years ago, a couple of years ago. She got a lot of vaccines at once and developed some paralysis type syndrome, which lasted a few weeks and then went away. This is okay. Is Beyond the Noise not on Apple Podcasts, only on YouTube? That's correct. It's not on Apple Podcasts. So it's only a video version. I didn't have any. You're only the second person that asked for it. Maybe you're not even asking for it. I don't know. But I just wanted to keep it video. Joseph is in Southern Ontario. Welcome, Joseph. Research about longevity from the blue zones suggests having community is another factor in longevity. Yes. There are other things that are factors as well. Yeah. So Wipeout. Why are we mentioning Wipeout? Surfin' USA. Surfin' USA was the Beach Boys song, right? Not Surf's Up. But Wipeout was a great song by... Who did Wipeout? Oh. And that's a game show song. It was the Surfaris. Wipeout. Remember? I wish I could play music clips here. I would have played that right now. This is better than me. It was someone laughing very eerily in the beginning, and then the crackling noise, and then they would start playing. Yeah. And Redondo Beach is a song by Patti Smith. Perpetual Sky wants to know if a virus could evolve to repair its host instead of harming it. So there are viruses that do not harm their hosts. They cause asymptomatic infections. But here's the thing. Viruses are parasites, right? They get into cells and they take something from the cell. So that's harming the cell, even though it's an asymptomatic infection. So we all have these polyomaviruses in us that reproduce all the time. And they don't seem to cause any problems unless you're immunosuppressed. Then they go out of control and you get disease. But in an immunocompetent person, they don't seem to cause a problem, but they're still taking away. So I don't see how a virus could repair if it's taking away stuff. That's the problem, because it's an obligate intracellular parasite. So I think by definition, it's always going to either be neutral or harm the host, but not repair it. That's my view. March 716, Redondo Beach is so lovely. I visited my cousins there a few years back. Polka Dot Pup is in Denison, Texas, 89F. I watched Dr. Offit on the Bulwark this afternoon. Excellent. My tomatoes are ready to eat. Anyone gardening this year? A lot of people are gardening here. Eric, the Richelieu Pondre thing wasn't meant to test your French. Hit send early by mistake. I don't mind at all. I took a lot of French in school, and I'm always happy to see how much I can remember. And I also took some Italian, but when I was a kid, between 1 and 5 years old, and I grew up in a house that was speaking Italian, right? My father was from Italy. My mother's parents were from Italy. We all lived together. So I learned Italian. So it's still in there. It's in there and I have to get it out. So I have a good aptitude for Italian, but I don't know anything. I don't know enough. I forgot it all. But if I went to Italy for 2 months, I could talk good. I could speak really well. Yeah. We had a professor in microbiology at Columbia, Hamish Young. He was from England, and he always spoke perfectly. And one year at a retreat, the students did a skit to make fun of some of the professors. And they had him starting a grammar company where you could call them up and, ask them about grammar. And the number was 1-800-TALK-GOOD. Isn't that great? Yeah, they're very clever kids. Molly writes, the internist I worked with in the 90s predicted that everything would be treated as infection. Well, not everything is, as you know, right? But many things have a basis in infectious disease, for sure. And maybe even chronic diseases, right? Certainly many chronic diseases, and now we see long COVID. But there has always been the suggestion that certain kinds of diabetes had a viral origin, not proven. But of course, hepatitis, chronic infection, and so forth. Polka Dot Puck read that alligator Alcatraz may have COVID circulating about. Okay. Christine is in Houston, Texas, celebrating my team's first phase 3 engineering purified exosome product intended for wound care therapy. Congratulations, Christina. Phase 3, that's great. Very, very good. Javier is in Southern Ontario, 24C. Hello, John in Minneapolis, 25C. How to choose your specialization. There are so many viruses. It's kind of, it's almost dictated. So I went to Mount Sinai in New York for a PhD. And I decided to work for Peter Palese. He's a young guy with a new lab, and he worked on influenza viruses. I thought that would be great. So then I got locked into influenza. Although as a postdoc, I could have done something else, but I found it fascinating. And then as a postdoc, I just picked the best RNA virus lab I could find, and that was David Baltimore, and he worked on poliovirus. So that's how I got to do what I wanted to do. So the lab plays a big role because you need to go to a good lab that's going to train you well and give you opportunities. I was very fortunate. So I didn't know anything about viruses. I just liked the idea and I wanted to work on them, right? So I depend on other people's advice. That's the other thing. You have to find people who will give you advice, but you have to listen to them. So you have to find good people. And a few people in the department told me I should work with Peter, that he was going to be really good, and he was. He did become great. And so I did. But yeah, that's it. You have to find mentors that you're going to trust. Elizabeth had a deer that probably has chronic wasting disease in my suburban backyard for a couple of these. Seems to have moved on. Any new data? No, the only data are that it's continuing to spread throughout the U.S. and Canada. Every year I talk about it in my course. I get a new map from the, I forgot the name of it's a government organization. And there's more deer that have been found to be infected. So there's no evidence that there's no human has yet contracted a prion disease from deer. Don't know if that's going to happen eventually or what. But you could go. You could go to my lecture from this year and get the scoop. It's the end of the prion lecture, and it's just like 10 minutes. But yeah, he was using sciencey word, phylum, universal vaccine. Yeah, didn't know what he was saying. John also liked immune booster. That was Shane Crotty. Very interesting to hear how they had all the right people and facilities to get a paper done in 7 days. Talk about burning the midnight oil. When you get scientists passionate, man, there's no stopping them, unless you take away your money. And that's a good way to stop them. Maureen is in a warm and humid Northeast Ohio. Welcome, Maureen. Betty K is in Washington Heights. So hard to watch the news these days. Yes, not just science news, all news, right? I mean, this nonsense with Epstein, this nonsense with meeting Putin. You're not supposed to meet someone in your country who has invaded another country. That's from World War II. And he doesn't have any sense of history whatsoever, or decorum, or humanity. Questella, we have West Nile in my county now, and I have about 18 bazillion mosquito bites. Fun times. Les writes, RFK's only qualification was getting votes for the president. Molly writes, high-dose vitamin C. I have to get IV weekly. Haven't been sick in 2 years. I used to get sick 2 to 6 times a year. Melinda writes, there are a lot of foods and drinks that fight COVID in a petri dish. Have any of these been studied in the body? No. You need to find the ingredient that is inhibitory. And now it's hard because we have some antivirals and it's costly to make new ones. And it's going to be hard to clinical test it. Christina's in Hershey, PA, where it's 25C and cloudy. Hello, John. Welcome. It's so friendly here. It is. It is friendly. Questella, people at one kid's school were bragging about not getting vaccines for chickenpox. What's that to brag about, right? Oh my gosh. So sad. Polka Dot Pup usually gets his flu shot around Halloween. Is that about right? Yeah, I think that's the end of October, right? October, November is fine. Yes. Barb Mack, I've actually got tomatoes ripening in my garden this year because it's been so consistently hot and dry. Molly hates missing this. It's always fun and educational. Sorry I missed it. No worries, Molly. You got other things to do. It's fine. But I'm glad you like it. We hear that Aussie flu precedes northern flu. It could work the other way around, of course. So it depends what you're centric. So here in the northern hemisphere, we say we get our flu from the southern hemisphere. And down there, they say they get it from us. Yeah. Barbara's in Durham, North Carolina, 85% humidity. Wow. Wow. School's out, but never stop learning. That's a good motto. I like that very much. Yep. Here, Peter is the Frank Zappa quote. The universe consists of 5% protons, 5% neutrons, and 5% electrons, and 85% morons. Hello, Rainey. Welcome. Peter, about COVID boosters. You might want to encourage people to get them to reduce chances of long COVID. 3 years with long COVID, I'm still messed up. So, yes, I should probably say that there's really good evidence that vaccines do reduce the incidence of long COVID, which is a nasty illness, as Peter points out. Yeah. Okay. Molly wants to know your dog's paralysis like Guillain-Barré. It's called acute idiopathic polyradiculoneuritis, neurological condition in dogs that causes inflammation and degeneration of the nerves near the spinal. It's similar to Guillain-Barré in humans. Yes. And she recovered. She was okay. But for a while, the rear legs didn't work. She was in pain, didn't eat well. Yeah. Very Nika. Speaking of personal stories of parents losing their kids to vaccine-preventable illnesses, today I found out it's the story of Benjamin Franklin. That's how he became a vaccine champion. Really, that's good. And don't forget, pull off its Substack. You can read about it a week before we publish the video. Visto, I think Beyond the Noise as a podcast could find more audience. It works fine as audio. It will be fine as audio only. Okay. I'll try it. Yes, Physics Girl was continuing to have improving health in her long COVID struggle, still significant challenges. Yes. Yes, in fact, Ben Franklin championed the smallpox vaccine and had his family immunized. Yeah. The doctor who did that died in the 90s. That reversed heart disease. Later wrote a book called It's Not the Diet, More Relaxation in Relationships. Wipe out great drum rhythm. Yeah. In the beginning. That was the whole melody. And it goes up, whatever notes. Yeah. MP Immune should have Dr. Andrea Love on us. I guess I'll run it past my co-hosts for sure. Kip, hello. Thank you for your support of science communication. Looking at the weekly wastewater graphic, will COVID ever decrease into the very low category? It never has, right? I don't think it will. I don't think it will. Okay, one more, and then we'll do some other things for a while. Visto, you know that rare morning you wake up and feel just great? That's when you caught a positive virus. Yeah, I agree. Okay, I want to do some news first. First item is an article in Science. It's entitled NIH Ponders overhauling the HIV budget to capitalize on prevention breakthrough. I think I can share this. There you go. So basically, lenacapavir, right? This antiviral that we talked on last TWIV is it great for preventing infection. You get it twice a year subcutaneously and it protects you. It's great. So leaders at the NIH are discussing the possibility of a massive shift in investment away from basic research and toward implementation science. Studies are the most effective way to use tools like the breakthrough drug lenacapavir. The agency may devote $1 billion, 1/3 of the agency's budget for HIV, to this approach. The idea has alarmed scientists both inside and out because basic research that drives HIV treatment, prevention, and cure would lose about $800 million annually. So it's an interesting article. It looks like it is open access. I don't think you should put all your eggs in one basket. I think you ought to diversify, remain diversified, and use some money to implement more widespread lenacapavir. Other countries need it too, besides us, but we don't support other countries' needs any longer in terms of health. It's a shame. But I would continue to do research because that's where innovations like this drug come from, right? So that's an interesting article. I like that. Okay. The musical chairs at the FDA. Vinay Prasad returns to role as top vaccine regulator at FDA days after leaving the agency. So, less than a week, a little more than a week, the FDA asked him to come back. Criticism of his tenure intensified around the agency's handling of a gene therapy for Duchenne muscular dystrophy. The FDA-approved therapy played a role in the death of 2 teens with advanced disease. After a third death, the FDA asked Sarepta, the company, to stop all shipments, changed course later. Laura Loomer, a far-right activist and an ally of Trump, posted on a blog on July 20th that Prasad was a progressive leftist saboteur who was undermining the agency's work. So get this. This left-wing, far-right activist writes something. The White House sees it, and they fire him without checking anything because she's a whack job. And they should never have listened to her. But although actually that's what this administration does. It employs whack jobs and listens to them or doesn't listen to them. Oh, my gosh. So there was really no basis for firing him based on Loomer's column, although I think he's not qualified for the position. And, saying you don't need to give kids COVID vaccines is absurd. Saying you don't need to immunize pregnant women against COVID is absurd. I don't think he knows what he's talking about. He says, there's a myocarditis concern. No, there's very little myocarditis concern anymore. Interesting, right? So basically, the HHS spokesperson, Nixon, and I forgot his first name, when they fired him, or they asked him to leave, or they said he resigned, he wanted to spend more time with his family. This was an outright, abject lie. It's just disgusting. That's what they do all the time. A couple more stories. You may have seen this one. So there was, of course, a shooting at CDC. The police believe that he was mad at CDC because he thought the COVID vaccine caused his health problems. So the gunman killed 1 police officer and shot multiple rounds through the windows of CDC headquarters. So this is an interesting post on this Substack called Weaponized by Carolyn Orr Bueno. And she writes, if the shooting was indeed motivated by anti-vaccine sentiment, it would add to a long list of anti-vaccine related violent incidents in the U.S. While the anti-vaccine movement has existed for many decades, it has recently become more organized and increasingly aligned with political extremism. Can you imagine it's the anti-vaxxers are aligned with political extremism? Of course they are. We've been saying that all along. But that's the danger of spewing lies about vaccines. At some point, someone is going to get pissed off enough to shoot. And if that, it still doesn't affect gun control. Now, did you hear not too long ago that RFK Jr. Said that Alzheimer's was a kind of diabetes? So here we have an article in the Alzheimer's Association website. Alzheimer's Association responds to Senator Kennedy's mischaracterizations about state of Alzheimer's research efforts. In his testimony, Secretary Kennedy said Alzheimer's disease is now characterized in the medical literature as type 3 diabetes. While there are, and then end quote, while there are commonalities between Alzheimer's and diabetes, labeling Alzheimer's as type 3 is inaccurate and misleading. While some evidence suggests that Alzheimer's and diabetes share similar underlying mechanisms, such as impairments in cellular metabolism, Alzheimer's disease is not diabetes. Calling insulin dysfunction in the brain diabetes obscures and oversimplifies complex diseases and risks misinforming the public. Later in the hearing, Representative Nanette Barragan from California, a long-time advocate for dementia research, questioned Secretary Kennedy about proposed funding cuts at the National Institute of Aging. When asked if he was familiar with the Alzheimer's Disease Research Centers, he said, "No, I can't say that I am." "This admission is alarming," said Joanna Pike, CEO of the Alzheimer's Association. Okay. Do we have any more news? That's it. That's enough. He just... he doesn't know anything, right? Now let's do a mini lecture. As I said, this was requested by you folks. DNA sequencing. So here we go. So the title of this is DNA sequencing, but that's actually jargon. DNA sequencing is jargon. What's correct is DNA sequence analysis, okay? But I'll use DNA sequencing because it's shorter. So what is this all about? So as you know, your chromosomes, here's one of your chromosomes here, right? This one, well, they all have two copies of the DNA. And they are made up of DNA, which is a double helical molecule. And it's all wrapped around proteins and stuffed into these chromosomes. We have billions of base pairs of DNA. But the DNA looks like this. It's a double helical molecule whose structure was solved by Watson and Crick in 1953. And it consists of a backbone. So this line that is going up and down, that's the backbone. And it consists of sugars and phosphates. So here, for example, at the bottom here, you see there's a ribose, this sugar, attached to a phosphate. And then there's a base attached to the sugar. And the bases on each strand of DNA base pair with the other strand. And there are rules. T only base pair with A, and G only base pair with C. So there are 4 bases. There's adenine, guanine, thymine, and cytosine, and they base pair specifically. And so that's what DNA is composed of, a whole string of these letters. And why is this important? Well, DNA specifies proteins, among other things. But the sequence of this DNA is organized in triplets, is read by triplets. So the DNA is copied to mRNA, and then the mRNA is read by ribosomes to produce proteins. And so these bases now, each triplet specifies a different amino acid. So that's why we want to know the sequence of DNA. We've wanted to know for some time. And it wasn't until the 70s and 80s was the technology developed to do DNA sequencing. And now it's just proceeded amazingly, as you'll see today. Now, why do we want to know the DNA sequence? Well, we want to know what genes are in an organism, how they change with disease, for example. We'd like to know the genes of humans and how they relate to disease. So lots and lots of applications of knowing, figuring out this sequence. So that's what we mean by the DNA sequence. Here, TAC, TCA, GGT, TCA, that's the sequence of this little stretch. Okay, so how do you do it? So I did one of the first viral genome sequences back in 1981. I was a postdoc with David Baltimore, and he told me to work on poliovirus. And since I trusted that he knew what was best for me, and he was right, I did. So he said, I want you to clone the genome and sequence it. So what does that mean? So you take poliovirus and you know, poliovirus is a nice compact capsid virus, and inside is an RNA. So I extracted the RNA from the virus particle. Now, you can't sequence RNA very easily, but what you can do is you can convert it to DNA and then sequence the DNA. And so what I did was make a DNA copy of poliovirus RNA. I put it in a bacterial plasmid so I could grow it and make more of it. And I sequenced. I determined its genome sequence. And so I had to convert the RNA to DNA. And obviously, if you want to sequence a DNA virus, you don't need that step. But for RNA viruses, you use reverse transcriptase to convert the RNA to DNA. Okay? And this is what I published on the right. This is part of the sequence of poliovirus. It's 7,440 bases. And you can see there's an open reading frame starting at base 742 and you can see the amino acids that are encoded by each triplet. It extends actually throughout the whole length of the RNA. So we knew what the protein sequence was of poliovirus, and we could nowadays you can see how it varies. You can use that sequence of many, many isolates to do evolutionary studies. You can mutate the genome and so forth. So having the sequence is great. So here's what I did to get this sequence. It took me 1 year to get this sequence. It would take 30 minutes today. I would just mail it to a company and they would do it in 30 minutes. But what did I do? I used the technique called Max M. Gilbert sequencing, which is a method that uses chemicals to degrade DNA. Now, it's not used anymore because these chemicals are toxic. It's been replaced by other approaches. But let me tell you how it works. So what you start with is your DNA on the left here. And you can't use huge DNA. So polio is 7,440 bases. I had to chop it up because that's too long for the sequencing. You can only get 400, 500, 800 bases of sequence at once, which is pretty good, actually. So I would cut it up with restriction enzymes and purify the fragments and have a fragment of, say, 800 bases in length. And then I would, so DNA, when you cut it with restriction enzymes, at the ends there's a hydroxyl and a phosphate. Here, the H was cut off, a hydroxyl and a phosphate. And we want to remove the phosphates because we want to replace them with radioactive phosphate, P32. Because back then, that's the only way we could trace sequencing was by radioactivity. So you would dephosphorylate the DNA with an enzyme. And then you would use another enzyme called polynucleotide kinase to add radioactive phosphate to the ends. So now you have DNA with a radioactive phosphate in each end. Now you have to cut this DNA because you can only have the radioactive phosphate at one end of the molecule. The sequence won't work if it's at both ends. So you have to cut it with restriction enzymes and then separate the fragments on a gel. So it's pretty laborious. That's why it took me a year. And then you do 4 chemical reactions. You take a little bit of each DNA and you put it in 4 tubes and you do 4 reactions. They're called G. So this chemical, I'm not going to tell you what the chemicals are, but one of them was piperidine, rocket fuel. It was very bad to have around. It never blew up on me, but oh boy. So this chemical cuts at G's. This one cuts at G plus A. This one cuts at T plus C. And this one cuts at C. So what you do is you take the DNA, which is labeled at one end, and you put it in these 4 tubes and carry out these reactions. And then you separate them on a very long, thin gel. So you get very high resolution. Basically, each band differs by 1 base. And so here, for example, and you're separating the fragments by size. So the top ones are longer than the bottom ones. So here, for example, we have a band in the T plus C and the C. So that makes it a C. If you have a band only in the T plus C, that makes it a T because the C is not reacting, right? Same with the G plus A and the G. So you could then read off these bases. And I would do this. I would run these gels. You would have to put a piece of X-ray film on them to expose them and get this autoradiogram, as we called it. And we would go, let's see, T, T, G, G, T, G, T, A, G, etc. I would just write it down on a piece of paper and then type it into a computer. Can you imagine the error rate? Oh my gosh. But I did that for the whole polio genome. So that's how Maxim Gilbert works. And this is the gel. This is Wally Gilbert. So he got a Nobel Prize for developing this method with Alan Maxim. And you would lay these, you would run really big gels with lots of samples, and you would lay an X-ray film on it, expose it for a while, and then develop it. And you could see all the bands. This is a crappy gel. It's very hard to read. My gels were great. I had hundreds of these gels. I threw them all out. When I moved to Columbia, I should have kept one to show you, but I didn't. So that's how it works. Okay, so even before Maxim Gilbert, another sequencing method was developed by Albert Sanger. And he got a Nobel Prize for this. He also developed protein sequencing methods for which he got a Nobel Prize as well. He was just bloody amazing. This is a enzymatic reaction. It's a primer-dependent DNA synthesis reaction using chain terminators. Okay? So the chain terminators are dideoxynucleotides. So our DNA is built with deoxynucleotides. All right? We have phosphates, sugar backbone, and the base is attached to the sugar. The DNA is deoxy-NTP. So there is a hydroxyl here and a hydrogen here. And the hydroxyl is used to attach the next base during DNA synthesis. Dideoxys, which are synthetic, have 2 hydrogens. So you can't attach another base to a growing DNA chain. It's a chain terminator like many antiviral compounds are. And so what you would do is you would take single-stranded DNA. It doesn't have to be labeled now. And you would incubate it with DNA polymerase, a primer, and dideoxy-NTPs. Okay? So you would substitute a dideoxy for one of the other. So you need all 4 DNTPs, and you would substitute dideoxy for one of them. And this originally was radioactive as well, but then it developed that you could make fluorescently labeled dideoxy NTPs, and so you could still separate them on a gel, but you could also use the sequencing machine to read the colors of each band and automate sequencing. But what would happen is if you had dideoxy GN, then the DNA sequence this polymerase would copy this until it hit a C, and then it would stop. And so with each of the dideoxys, you would stop at different places depending on the sequence. And again, the gel separates according to size. So you can simply read off the sequence by looking at where DNA synthesis is terminated. This is still used today because, as we'll see now, there are high throughput methods of DNA sequencing where you can do lots of samples at once in a very short time. But they're error prone, and you can use Sanger sequencing to confirm those sequences. So these were first generation sequencing methods. Then the real revolution began with the second generation around 1996. These were methods based on what we call pyrosequencing. And the very first one was the 454 methodology developed by Roche. And this was actually a machine like this one that would pretty much do everything for you. And everything I'm going to explain what it is in a minute. So these kinds of sequencing, these second generations, are fast. So you could do poliovirus in a half hour. And they're parallel, massively parallel. You could sequence multiple samples at once. When I did poliovirus, I did only one virus because it was too laborious to do more than one. But with these machines now, you can do many, 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 many different ones. So how does this work? What is pyrosequencing? So here's ATP. It's one of the 4 bases that's incorporated into DNA. You buy it as a triphosphate. So here's the base here, adenine, the ribose, sugar, and 3 phosphates. When this is incorporated into DNA by a DNA polymerase, the last 2 phosphates are cut off and released. So there's just 1 phosphate between the nucleosides in the DNA. These 2 are called pyrophosphate. This compound, POPO, is called pyrophosphate. It's released whenever a base is added to DNA. So basically, when a polymerase is copying your DNA, every time it adds a base, some pyrophosphate is released. And they figured out a way to measure that by light, by measuring light. So when the pyrophosphate is released, there's a cocktail that's used that contains enzymes that will use the pyrophosphate to make ATP. The ATP will then drive the production of light by luciferase enzyme. So the pyrophosphate release can be measured by measuring the light at each step. So you don't need radioactivity and you don't need dideoxy. You just are measuring the sequencing happening at each step. So that's pyrosequencing. But the way it actually is done in the machine is amazing. So you start with DNA. You break it up into small fragments and you make them single-stranded. Then you do PCR in an emulsion in these oil droplets in water. And each droplet has a bead. And the DNA is attached to the bead and it's PCR amplified on the bead. So these single-stranded DNAs would be attached to the bead. And then you do a PCR reaction. And then each bead ends up with many, many, many copies of the same DNA. Okay? You then take these beads and put them into wells, very, very tiny wells. And the machine does all of this. And then you carry out a PCR reaction. Sorry, the polymerase reaction that I just talked about, where we measure the light emitted with each pyrophosphate release, and that's measured by a machine. So you basically, the way it's done is the polymerase and the primer attached to the DNA on beads. The bases are added to the wells in waves of 1 base at a time. That's why you have this machine with, tubes of A, T, G, and C, and one at a time will get put through A's, C's, G's, and T's. And when each base is incorporated into the DNA, light is given out. So, you put T in and maybe there's no T there at the end. You put C in and maybe there's a G that templates. So now you have a C there and you get the pyrophosphate release. So it's recorded. And then you switch bases being added and you get something else. So it's all polymerized on the same template in series. The intensity of the light that's released corresponds to the number of bases that have been incorporated. So if there are 3 consecutive A's, the amount of light would be 3 times that of a single A. And if you plot the light intensity on a graph, you get the sequence of the DNA. So that was revolutionary, as you might expect. You just would do your PCR reaction and then give it to the machine, and the machine would take care of it. In fact, maybe the machine would even do the PCR reaction. It would spit out the sequence. Now, that isn't used anymore. It's too expensive and it's just not good enough. But other technologies were developed that were based on different approaches. And here there are 2 called Ion Torrent and Illumina, 2007 and 2011. This is called sequencing by synthesis. And here, instead of detecting the pyrophosphate, they detect hydrogen ions when the DNA is made. I mean, 454, Roche had the patent on pyrophosphate, so nobody else could use it. So they figured something else out, and they used hydrogen ions. So what you do is you take your DNA, you break it up, and then you add adapters to the ends, which are basically barcodes to identify each DNA. And so that's going to be your sequencing library. You're going to attach the DNA to beads, just like you did before, and do some PCR to amplify it. And then the sequencing reaction is done in a well, just like before. And you're putting in different DNTPs, just like we did before, but we're measuring the release of hydrogen as each base is added, because not only is pyrophosphate released, but hydrogen is released, and that becomes a blip on the graph, and eventually you can get a sequence when you use the different bases. Okay, so that gives you kind of short reads, and then you just overlap them by computational methods to get the final assembled sequence. So that's why we talk about these next generation sequencing. We're always talking about reads, and that's just, you have a piece of DNA that you've put in, and how many times do you get the read, the sequence of it? So then we have in 2012, this is very cool, Oxford nanopore sequencing. And this one measures changes in electrical conductivity that occur when DNA strands pass through biological nanopores. And so here in the machine is assembled a membrane with this nanopore in it. These are biological nanopores. And on top is a motor protein. And you add DNA, and the motor protein will separate the strands and put one strand through the nanopore. And there's a charge on either side of the membrane. When each base emerges, it has a characteristic drop. It causes a characteristic drop in the ionic current here. So each base gives a characteristic reduction in the ionic current, allowing the DNA to be sequenced. It's so brilliant, all of these approaches, right? It's so amazing. So that's this is the one that is being used a lot now this year. And then we have PacBio single molecule real time, which is also cool. So I wanted to show you this. You take your sequence, your DNA that you want to sequence, and you put adapters on either end. So now you have a circular DNA, and you put it in a well where the polymerase is attached to the bottom of the well, and the polymerase will basically keep copying this DNA over and over again. So you get lots of reads from the same DNA. And what happens is you use fluorescent nucleotides, and the fluorescent intensities are measured over time, and that gives you the sequence. So read lengths are very important here. How far can you get? So some of these techniques like Illumina and sequencing by synthesis, you get short reads, 50 to 500 base pairs. So you have to get lots of them and overlap them on your whole genome. Some of them do long reads between a kilobase and a megabase. PacBio, Oxford Nanopore can do that. And the older technology, Sanger sequencing, could do 500 to 1,000 base pairs. So that's not bad. And we still use Sanger to confirm some of the results of these other read technologies. Data output per run. So in gigabases of DNA, with Sanger, it's very low because you have to run a gel and you get 400 or 500 bases if you are lucky. But with pyrosequencing, ion first generations, and then ion torrents, Illumina, you could get more and more, and of course the long reads more as well. So we've improved the time it takes as well as the data output per run. What's amazing is that this has now become a huge market, right? I forgot what CAGR stands for, capitalization of something. But here in 2022, the market for next generation sequencing was $3 billion. So this is not just research labs, but clinical labs who want to sequence patient DNA and so forth. And you can see all the applications here: oncology, sequence your tumor, reproductive health, clinical investigation, consumer genomics, HLA typing, metagenomics, epidemiology, drug development, agrogenomics. They're all shown here. Oncology is obviously a big one. So is reproductive health. So is clinical investigation, consumer genomics. Metagenomics and epi would be sequencing, COVID and flu viruses. But look, it's predicted to go up hugely by 2030. So it's financially, it's a huge market because we all want to know genome sequences, right? So that is sequencing. It has come from being really something that you only did in a research lab. I did. It took me a year to sequence poliovirus or genome, to now it can be done in 30 minutes, and you can do 1,000 different poliovirus sequences at the same time if you wanted to. And it's fast. We can do human genomes. We can do animal genomes. We can do viruses, bacteria really quickly and get a lot of cool information. So I think it's just amazing. And thank you for suggesting we do this because it's very cool. And we also have a request for PCR technology, which has also evolved from the early days. And I'll see if I can get that together for you next time. Let's see if we have some questions here. Tona wants to know about awakening breast cancer cells. I know that they looked at respiratory infections. Wouldn't it be likely that other inflammatory producing effects would likewise wake up? It's possible, yeah. But the thing about breast cancer is that the metastases are in the lungs. They're dormant. So according to your oncologist, you're in remission. The CAT scan shows no masses, but there are cells there, which you don't pick up on a CAT scan, right? And then you get a respiratory infection, and the inflammation is right there, right? But let's say you had a leg wound and you had inflammation in the leg wound. Would that reactivate a lung dormant cancer cell? I don't know. I mean, cytokines that are produced during inflammation certainly move around the body, right? So it's conceivable. We talked about this on the TWIV. We discussed it, but I just don't know. The experiment needs to be done. And also whether other cancers form dormant cells like this and whether they could be reactivated as well. Joseph is here. Buonasera, professore. Buonasera, Joseph. I'm just learning Nevitz writes about ultra-conserved elements, short segments of DNA showing nearly 100% sequence identity among human, mice, rats, which wish Twixzy could cover it. Let me write it down. I do have these notebooks I'm supposed to use to write stuff down in. And so I'm going to do that. This one is pristine. Look at that. Let's write it down. Where's my pen? Come here. UCE. Oh, it's so nice to write on this paper. Ultra conserved DNA elements. Okay, we'll do that. Did you like the Vincent Price spooky intro to mystery? Yes, very much. Also liked how Rod Serling would introduce his show. Hello, John from Port Townsend, Washington. 66F. Yesterday was summer. Christina was also inspired by a French-speaking family to learn in school, then Spanish. I can understand both well at work. Very good. Barbara says, I always hear about the incubator, but don't really know what it is. The incubator is our recording studio in New York City on 7th Avenue. I'm not there now. I'm home because these are late live streams, and it's just very difficult to get home at this hour at 10 p.m. I used to do it, but it was unpleasant, let's say. Anyway, it's in New York, and we record pretty much everything else there. And it's a lovely space. If you're ever in New York, come visit. We're very lucky to have it. And we call it the incubator because, an incubator is a piece of equipment in a lab. And of course, an incubator is also where you develop ideas. It's perfect, right? John donated via the link on the screen for another great week of content. Thank you very much, John, for your support. Really appreciate it. My family, Tona writes, knows I need my alone time on Wednesday nights because we have a date live with Vincent Microbe TV. Yep. Molly watches this for her sanity. I love facts and data. Any thoughts about Legionnaire's disease in Harlem? Daniel talked about this last week. Quite a sizable outbreak, likely caused by cooling tower water, which harbors the amoeba, and the Legionella live inside of them. But they're going to have to find out, where the reservoir is causing that. And Daniel wasn't very hopeful. He thinks it would just burn out on its own. Costello says she had to stop watching most things because it started so many fights. I'm sorry about that. Abdullah wants to know, have we accepted prions as their own organisms? No, they're just proteins, right? Not organisms. So they are certainly pathogens, but they're your own proteins that go bad, that go wrong. I remember do you remember Wallace and Gromit? There was a penguin. They took in a penguin, and the penguin was a crook, basically. And at one point, Wallace said, he's gone wrong. He's gone wrong. So it's prion is a normal protein that's gone wrong. Daniel from Brazil, welcome. Brasilia, which is in Brazil. Lovely. Nice to have you. 23C there. Thank you, SRR, for your contribution to science communication. We appreciate it. We need your contributions. Yeah. Love it. An ounce of prevention is worth a ton of cure. Aubrey, good evening from Richmond, Virginia. Rainy 26C. Remember, if you take advice, don't blame the advisor. No, that's why I say you have to find someone you're going to listen to and listen to them. But then you cannot blame them because things will go wrong. Oh, I had all kinds of trouble in Peter's lab. Just have to work through it. It's not his fault. He said, you should do this experiment. I said, it's a good idea. And then I can't get it to work. It's not his fault. Yeah, that's totally good advice. Charlotte got the flu shot and COVID shot today because school starts on Tuesday and I'm teaching 50 students in one classroom. Good luck with that. Tryon says, when Rouse studied virus-induced sarcoma, it seemed that starving mitigated infection. I don't know about that. I'd take your word for it, but I'd have to go look it up. I don't know why that would be. Because tumors have a high metabolic demand, and they actually do anaerobic metabolism, like glycolysis, taking sugars and making lactic acid. So I'm not sure how that goes with that. COVID is not a new one. It's the one we got a year ago or 6 months ago. It's the same one. They didn't change. It's fine. Daniel says he's not convinced that there's any difference in having the newer one. Yeah. Tryon says maybe low zinc levels decrease virus replication. Some viruses, their enzymes have zinc cofactors, so that would reduce their replication. But it's not very useful therapeutically, right? Michael writes, the news about science funding is so depressing. I've never had any qualms about paying my federal taxes, as I knew it was going to things like NSF or NIH. Yeah, I agree. Julie S. writes, in the 80s, I was a non-vaxxer until my 4-year-old daughter got whooping cough and almost died. This changed my mind. Well, Julie, would you be interested in recording like a short interview with me where you talk about that we could use? I know if you don't want to do it, that's fine. But if you're interested, we can do it online, and just shoot me an email, vincent@microbe.tv. I would love to get stories like yours put out there. So to make it personal, as someone was just saying before, right? Thank you so much for telling us that, because I'd like to know what you were thinking before and after. Polka Dot Pup, have had 3 COVID shots. I wasn't going to get one this year, but I'm concerned the government might completely remove them. I don't think they're going to completely remove them. They're just going to be making it difficult for you to get them. The companies will still make them. Christina writes, employers are no longer sympathetic about long COVID employees. It isn't considered an ADA disability yet. One gal lost her job due to long COVID symptoms. Yeah, that's not right. Visto, if you play old Greek bouzouki records at high speed, then you get 60s surf music. You know, I have an old Greek vinyl bouzouki album, and I can crank my turntable up to 45. I'm going to try that. Yes, to be precise, Ben Franklin lived before vaccination. He championed variolation, which had already been developed by Edward Jenner, right? Greece is the cradle of civilization and rock and roll. I'm going to Greece in September. I'm going to Athens and then Thessaloniki for a meeting. Should be fun. Michael continues, now with my money going to things like ICE rather than science and public health, I'm going to hate doing my taxes. Eric writes, mitochondria are good obligate intracellular parasites. Some sea slugs have chloroplasts. I don't know if they're parasites anymore. They're giving us stuff, right? But they, it's a good question. We were discussing that somewhere before about whether maybe Mark Martin on a twin, whether mitochondria are parasites. I suppose they do take things. They take reagent, but they give back a lot of energy, right? So it's a good deal. But you're saying that just because a virus is an obligate intracellular parasites, that doesn't mean it can't be good. But I haven't seen one other than mitochondria that and chloroplasts. Um, I haven't seen a virus that helps you in any way, although they might. Okay. I'm not saying that that's not a thing. We just don't know about it. Wipe Out the Ventures saw them live in 1969 opening for Jefferson Airplane. Wow. That's cool. I think they're a one-hit wonder, right? I don't know any other song that goes through my head like theirs. I had the 45, the little guys. Hmm. Prasad didn't exit in the revolving door. Yeah, he went around and came back again. Michael writes, people just don't get that this country's scientific community and funding apparatus really was a major part of American exceptionalism. It won us World War II and then powered our economy past the USSR. Yeah. They think that they can do it their way, but they are seeing, the economy is just horrible right now. Prices are rising, right? Mm-hmm. Anti-vaccine guns, what could go wrong? Sure. It says it all, right? RFK said he was going to find a cure for autism by, I think it was September, but soon, yeah. Well, yeah, I think he said he was going to find the cause of it. So we'll see what he comes up with. Paul Offit just said this week he thinks it's going to be aluminum adjuvants. It's going to manufacture some bogus study. Visto is not herpes virus reactivation associated with some 30% Alzheimer's. So RFK blame anything but vaccine preventable causes. That's right. Because he doesn't think that viruses cause disease. He thinks vaccines cause chronic diseases and we have to get rid of them. You know, he doesn't know anything. Why would he even have an opinion that matters? Why would anyone listen to him? He knows nothing, zero about science. That's what kills me. I mean, I've known much, much smarter people in science than him who are, sometimes they get things wrong, but at least they know their science. Oh my gosh. Oh my gosh. Alzheimer's as type 3 diabetes has been a crank notion that's been around a while. Not surprising that RFK Jr. sells it. That's right. Visto writes, the packing and unpacking of light years of DNA is amazing. How can it work? There are lots of places on each chromosome where the DNA polymerase sits and copies, but it takes a long time to do it. I forget what the number was. It takes days to copy a cell's DNA. So, no, it can't take days because cells can divide every 24 hours. It takes many hours. But, it's unpacked locally. So DNA is wrapped around discs called nucleosomes, and then those are packed together so they can get unwound a little for the polymerase again. But it definitely is amazing. And amazing that it works. And, everything has to be Goldilocks in you to get everything to work perfectly. And then it's not always Goldilocks, and that's when you get sick. Dr. McFarm, I understand that DNA sequencing and DNA sequencing analysis are chemistry and bioinformatics. Yes. Such as how bridge amplification is chemistry and short read assembly is bioinformatics. That's right. The amplification is chemistry. The short read assembly, you got your little short reads, and you let a computer find the overlap between them and make sure there are no mistakes in making the overlap. Yeah, the guy, Leo writes, who won the Nobel Prize for PCR, Kary Mullis, came up with the idea driving to the Mendocino coast one night. And unfortunately, so he was a bit of a whack job. I sat next to him at a dinner once. He's just crazy. And also ended up saying HIV does not cause AIDS. Just I don't understand this. There's overwhelming evidence. I just don't get it. Christina, that would have been a crazy fun lab sequencing polio RNA. I did it myself. I had some people around to talk to, right? And the whole floor, of course. We had lab meetings. It was good. I learned a lot. And it was a project. I completed it. I had never done anything like that before. I was able to organize all the biochemistry, as we just heard, and all the bioinformatics, which was pretty rudimentary. There were no programs to overlap the contigs, the little reads. We didn't call them reads back then, but the bioinformatics people ended up calling them reads. I did it by hand. So you would have 2 pieces of DNA, right, that you would sequence. And then you have a third piece that overlapped them so that you could say these 2 go together. And here's a story for you. So for every piece of DNA, 2 pieces of adjoining DNA that were sequenced separately, you had to have an overlap. I had hundreds of fragments across the polio genome that I sequenced. And I sequenced across all of them, but 1. I said, screw it. Actually, it was very hard to find an overlap. Damned if that didn't bite me in the ass, because a year later, when another lab did the sequence, they said, hey, Vinny, there are 30 bases missing from your sequence. Do you know what happened? So the enzyme that I used to separate the 2 fragments actually cut twice within 30 bases. And it was in frame. The amino acid, the DNA code was in frame, so I never picked it up when I translated it. Otherwise, it would have caused a frame shift and a termination. A perfect storm to make me make a mistake. Oh my gosh. Vanity wants to know what do YP3 and YP1. I think it's VP3 and VP1, the capsid proteins. We had done some capsid protein sequencing, so we could say, this is the VP1 gene, this is the VP3 gene. Molly says, I am impressed that I didn't know you'd done that. Oh, wait till you see what else we did. That was one thing that was very cool. There were 2 other things we did that are very cool too. We identified the cell receptor for polio, and then at a time when nobody had receptors, and then we made transgenic mice with those receptors and showed that they were susceptible to poliomyelitis. My lab was just cranking. It was great. Nevitz, for human DNA genome, 2% of the DNA sequence codes for protein plus, maybe a little less, some for different kinds of RNA. The remaining 98% was considered junk no longer. Tom, MG, Maxim Gilbert. So it's pretty laborious, a coy understatement. Those big, thin gels. Oh, they were tough. Very thin. Then you had to get them apart, the 2 glass plates, sometimes 3 feet long. You had to get them apart without breaking the gel. You've now done all these reactions, which have taken you a whole day. You have run them on this gel, which takes a long time. You open it and then the gel breaks. You could cry. You could bloody cry. Oh my gosh. And then you have to put an X-ray. You have to put a piece of paper on the gel to peel it off the glass because you can't stay on the glass. You have a big filter paper. You would put on, peel it off, then you would dry it and or put a piece of saran wrap on it and then put a piece of X-ray film on it and expose it at minus 70 degrees. That's why you couldn't have the glass in there. How big was the computer? So it was the MIT mainframe. I don't remember what it was at the time, 1980. Someone could look it up and find it, whether it was a digital equipment something or something else. But I had to access it through an acoustic coupler. You know what that is? You take a telephone and you put it in the acoustic coupler, which is linked into a local terminal, and then you log in remotely, and I would type in the sequence, and it would get sent over there. Oh man, the stuff we had to do 5 miles uphill in the snow. Ay, ay, ay. M. Rosa says, you also play with concentration, so the rate at which the dideoxys is rarely added so that you can get all the different lengths. That's right. Sanger sequencing with S35 alpha labeled NTPs was a huge release. Then fluorescent bases and instruments. And then, yeah, it's very cool. Pyro sequencing, yeah, 454, it's not made anymore. I mean, Roche bought up the company, and then they discontinued the product because there was better stuff. Nanopore sequencing. Oh, well, we said nanopore can do up to a megabase. You can put a lot of bases through that little pore. Isn't that cool? Legotic is going to change all my socials names to motor protein. And the next band name needs to be Megabase. Yeah, they're mind-blowing methods, aren't they? DNA consists of 2 parallel backbones, but we have read only one. So for Maxim and Gilbert, you would actually sequence both strands. You would get 2x coverage. Nowadays, you get 500x, 1000x, which means you get 500 reads of each strand, right? And it can be, depending on the technique, it can be either strand, but you need to get both strands so that you got some self-correcting there. And so I did that in Maxim Gilbert by sequencing both strands. Because the sequencing, you could read, in the old days, you could read the gel and make a mistake reading the gel, okay? You could get a compression or something, some artifact, and you get the wrong base. So if you do the other strand, you can fix that, yeah. I mean, the Oxford miniaturization, the Oxford nanopore is a little device the size of a harmonica that you do the reaction in, and you plug it into your computer, and it just spits out the data. It's bloody amazing. It's really amazing. The stuff we can do is just amazing. How did I purify the fragments? Run them out on a gel. Expose that briefly to film to see where the bands were, and then cut them out. Google says a full human genome costs $200 to $400. Okay, TP Steve wants me to explain the furin cleavage site thing again and why the R codons are so rare and why it isn't a smoking gun. That's a whole mini lecture. I'll do it next time, another time. Hey, H. Zhu, thank you for your support of science communication. I'm slightly surprised Ion Torrent has a seat in history of NGS. It was good for short reads. Yeah, I wanted to be complete. Basically, so Ion Torrent and Illumina turned out to dominate, right? It's the same sequencing by synthesis, detecting hydrogen ions, right? But the platform are totally different. I didn't really go into the platform here, but the Ion Torrent and the Illumina platforms are totally different. And that's what you have to do, of course, if you want to patent something. Pen Click has arrived. Who wants to talk about sunshine? That was last week. So the separating of the DNAs, it depends how big they were. You know, we would use acrylamide or agarose, yeah. John's upstairs living room becomes an office hours domicile Wednesday evenings. People know they enter. It is virophile time. I love that. Visto says prions are the bad proteins, the naughty ones smoking cigarettes behind the school shed. I like that. I like that very well. Oh, here we go. There it is. Thank you, Angry. The Wallace and Gromit episode with the penguin is the wrong trousers. It's the wrong Oh, so they put, the penguin puts Wallace in these trousers that walk, and then the penguin reprograms them. And as Wallace is saying, they're the wrong trousers and they've gone wrong. Ah, yes. Thank you very much for believing. Mm. What do I think about the oral COVID vaccine booster? This must be an adenovirus in an enteric-coated capsule, right? I haven't seen the data. I don't know. Are there data out there? You're in the clinical trial. That's cool. Why is it on pause? John writes, long COVID awareness helped me see that post-acute sequelae existed before for other diseases and deserves a voice too. That's very true. We had such big numbers with COVID that now you had to believe all the other ones, right? And it all happened more or less in synchronous, right? Angry Penguin, regarding Legionella, every time I climbed to the top of a cooling tower at work, I would practice shallow breathing, just paranoid, even though we use biocides. Yeah. Yeah. Not all commensals are parasites. That's right. That's true. Yeah. What is the actual concept of organelle? I would just say it's a subcellular structure with some function. Molly has had Lyme for 30 years, 50 grand a year. I'm so sorry, Molly, but at least it's under control. Nika wants to make humans with chloroplasts. Why do you want to do that? Does he want to be green, like the Hulk? Yes, Greece may be a cradle, but there were civilizations in Africa and Asia before Greece. Totally agree. Yeah. Franklin died in 1790. Jenner published Vaccination, 1798. Okay, we're wrapping it up here. Let's see if there's anyone else we should thank here for donating. A lot of great questions here. Bring them back next week. So many good questions. Oh my gosh. Wish we had another hour. Yeah. PDP-11. Might have been a PDP-11 with a modem, 300 baud modem. Yep. In 1980, the mainframe would have been an IBM 3033. Family, you're a living legend. Did Scientific American do a feature on your methods? No, they didn't. Nope, it's okay. I remember acoustic couplers. 5 miles uphill in the snow is an apt comparison. Oh my gosh, someone remembers those. That's great. Okay, lots of people lots of people are understanding what I was talking about, the old tech. Okay, folks, time to wrap it up. I want to thank our moderators tonight. We had Barb Mack, who's still here. It's great. It's late. We have Vanity Nutrition, Les, and Tom. Thank you very, very much for moderating tonight, and thanks everyone for coming. I really had a good time. I think you liked the mini lecture, and we'll do more. The next one will be PCR, so now we're getting to the part of my career where I didn't do very much PCR. I only did the initial generation PCR, but it's evolved substantially since then. So we will try it. Anyway, so I will look up what mainframe was at MIT in the 1980s. Is it a PDP or an IBM 3033? I don't remember, but it would be interesting to know. Thanks, everyone, for coming. We'll be back next week. I love to do these. I love to teach you guys what I know and what I've done. It's a lot of fun. And these are all recorded, of course, so if you can't make it, you can always listen later, and you can see the chat. We'll be back next week, August 20th. Boy, August is almost a wrap. Until then, folks, stay curious and be safe. Bye-bye.